Good afternoon, everyone. Thank you for coming for this IGHI student project competition. So this is really a third in our series that some of you may have attended in our previous ones. And every year we have increasing number of submissions. So this year, out of all the submissions, we have shortlisted 6 of them. And today we have a distinguished panel of judges. So Richard Smith, who is going to chair the judging panel. Richard, you all know that from his role as the editor-in-chief for BMJ and also CEO of the BMJ Group. He now runs the United Health Coronary Disease Initiative. And we also have, very honored to have the rector of Royal Corriva, Paul Thompson. He's also having this joint venture with Imperial. He is the co-director of the Helix Center. You may have read from the news last week that was opened by His Royal Highness Prince Charles. And we have here also from Imperial Innovation, Catherine Owen, that got this career in graduate from Birmingham, went to Manchester, then Imperial, now is part of the healthcare venture within the Imperial Innovation, and also her colleague Inga Deacon, and graduated from Cambridge, then went to Oxford, and also runs the same group. So this is really great to have all the the students and also the finalists here. And in your pocket, you also should have received a banknote, that £1,000, because you are not only here to listen to the finalists what they are going to present, but also you are one of the judges as well. So our judging panel, they are going to decide the first prize and the second prize, and the third prize will be very much left to you, to all of you, to decide who will be the winner for the third prize. And we'll bring in some ballot boxes, and towards the end you can put your vote into that. So without further ado, let me introduce Richard Smith to kickstart the competition. Richard.
Right. Well, good afternoon, everybody. So this is a Dragon's Den format. These are the dragons. I'm a dragon. We've got 3 others. Breathe a lot of fire. I assume you kind of know what the Dragon's Den format is. We're going to have 6 presentations of innovations, and we're going to be very, very strict on timing. So they have 10 minutes. We'll cut them off if they go a moment longer, and then we will grill them for 5 minutes on their presentations. And the criteria we're looking for are: how well does the project address a global health challenge? How innovative and creative is it? How will the money be spent? And we're going to, every competitor needs to tell us that. What are you going to do with the money? And how are you going to make sure that the £5,000 or whatever it is really has a big global impact? We're looking at the financial feasibility and sustainability, and 2 of the dragons here are global capitalists, very interested in how you're going to use the money. And we're going to look at the quality of the presentation. So let's kick off straight away. Joe here is going to keep the timing. As I say, we're going to be very strict. So the first one is VD Meta. So it's ILIAD, the International Laboratory for Identification of Antibacterial Drugs. And thinking up your acronym is always the most important thing. So well done.
Okay. Good evening, everybody. It's a real pleasure to present the ILIAD project before all of you today at the Global Health Innovation Challenge. ILIAD, as he mentioned, is the International Laboratory for Identification of Antibacterial Drugs. It was set up because of the lack of time and money the pharmaceutical companies are putting in development of new antibiotics. The goal is to search the globe for new and naturally occurring substances from plants, insects, bacteria, fungus, and other organisms using simple tests. To make this possible, we've developed the Postbiotics Kit. The toolkit contains all the equipment that enthusiasts can use to participate in the ILIAD project. Sorry, didn't realize. Okay. So that's the ILIAD project. Why? So antibacterial resistance currently accounts for an estimated 50,000 deaths in US and Europe alone. A recent project done by Wellcome Trust estimates that the actual current toll of death is 700,000. World Health Organization, in its 4-point strategy, mentions that promoting the development of new antibiotics and developing new diagnostics for test of resistant bacteria can assist us in prevention of drug resistance. Scientists are working to develop new strategies to combat the growing threat of germs that the current antibiotics cannot fight. So on the picture out here on the top, you see that researchers are testing substances such as silver combined with antibiotics to boost their killing power. Other researchers are also making use of genetic sequencing of bacteria to see how they can be formed into new drugs. Unfortunately, there isn't enough funding in this industry. Traditional drug researchers are housed in big pharmaceutical companies, which are funded by shareholders. Shareholders have vested interest in development for drugs for chronic diseases. And this has led to pulling out of funds from antibiotic research. This has further linkages in how national healthcare system and insurance companies are viewing and funding the antibacterial diseases. What ILIAD is proposing is to involve bioenthusiasts, DIYers, and school children in the development of global database of substances that may potentially have antibacterial values. In return, this raises awareness for the larger issue of drug resistance and can be an effective way of communicating the importance of prevention of infections and using today's antibiotics more effectively. We are Joshua Zaina, who's designed the biology for this kit and is currently supplying the lab equipment for all the projects. There are experts from University of Chicago who deal with the verification and further study in case a potential sample is found out using this thing. And I'm the designer who's helping to make the kit more user-friendly to reach out to a broader audience. So ILIAD was set up in December of 2013. And since then, we've kind of cleared lots of small milestones. We've developed an online platform for data collection. We have conducted workshops with these kits in USA and India, and we've created a kit for sale online. Our goal in the next 6 months is to see how the postbiotic kits can fit into the GCSE curriculum right now and create a crowdfunding campaign to manufacture these kits. The kit in its present version contains these components. However, we are coming up with newer methods in which the number of components can be greatly reduced, which can make it possible for easier process and reduce the chances for contamination. How do you test this kit? You collect the sample that you wish to test, you grind the sample, and you test it for antibacterial attributes against non-pathogenic E. coli bacteria. Results are visible usually within 24 hours, and you capture them photographically and upload it to the ILIAD web page. This is me trying out the kit for the first time. I'm no biologist. This is some of the children some of the grade 11 children in India testing the kit, so in the first beta testing that we did with them. They uploaded their results in this fashion onto the web page. I'm not sure if the projector quality is good enough, but if you see, there is a little white ring out here, and that indicates the antibiotic properties being indicated by that sample. So this is the web page that we have created using the SynBiota platform, and this is how we record each sample on the web page. We have also decided to locate the GPS and latitude and longitude, the scientific name of that substance, if known, and the colloquial name of the substance, if known. This will help in creating an efficient logbook which can be used further by the researchers. This is the online platform where we are selling these kits right now. Each kit is available for $60. This is an individual kit which allows person to test for 40 to 50 samples. We've also created school kits for $200, which allows a batch of 30 children to perform experiments. We're now developing a cell phone application that instructs and as well as allows to document the process as it's being performed, which makes it easier to be uploaded online. A key part of achieving success is enabling positive reinforcement for each result, whether it's positive or negative. We're trying experiments with staining the bacteria with colored and phosphorescence dyes. This will enable using computer intelligence to take photographs, and the photograph can recognize if there is any antibacterial properties that are found or not. We need funding to move this project forward. I'd like to end with the statistics on predictive deaths due to the drug resistance by 2050. If you see, the largest circles are around Asia and Africa. Ironically, these are the countries that have the most number of alternative remedies that are claimed to have antibacterial properties, like herbs, garlic, honey. Let's give the fight against antibiotic resistance a new hope by supporting ILIAD. Thank you.
Generous round of applause. You seem to have hit the point. What I'm going to do, I'm going to get each of the judges to ask a question, and then I'm going to mop up. Go, Catherine, you're first.
Just thinking about, obviously, tackling the global drug resistance problem is a massive issue. So it's not necessarily one approach is going to work.
Yes.
And it's all about building a community. So I just wondered, have you engaged with any potential pharma partners to start to build a network that you can tackle this together?
So we've considered many stakeholders. Schools is one of them that we thought is a much more approachable audience because they understand, there's like positive engagement with them because they're getting return on what they are investing into the kit. There's learning happening. With farmers, the problem that we had when we initiated conversations was them understanding the value of what do they get in return of this sort of an experimentation. And I think one of the way of tackling that would be to have chemistry involved, which gives them feedback on their own crops based on this thing. So then that would enable two-way interaction by this kit. But yes, we've spoken to educational institutes as the first step to make this possible.
What would you do with the £5,000 if you were to win?
For right now, our immediate goals is using it for development of a curriculum which fits into schools right now. So grade 10 syllabus, and how does this fit into the chapters of the existing curricular system. That's how we'll use this money.
Do you want to ask, that was a short answer. Do you want to ask another question?
Not quite yet. I'll save my fire.
Okay.
Just on that bit around the kit, what's to stop the school just building their own kit? Because I guess you've got quite a lot of common ingredients to the kit. So what is the benefit for them to work with you?
So I think that's where my design instincts come forward. I'm sorry I forgot to show this before. So
maybe you can sell one to Catherine.
So what we're trying to develop right now is, instead of having these multiple number of components that are in the kit and then you having to figure out anything, can it be one box with just one slide which you insert and then you see the results? Mm-hmm. It kind of makes the whole process much easier, and it also avoids contamination or anything else wrong happening to it. So that's what the version 2.0 is going to look like. Yeah, that's where it is.
Inga?
So where do you see this project going? So what does success look like for this project? Is it engagement of huge numbers of students, and I can see a huge benefit there, and education, inspiring new generations of scientists?
Yes.
Or is it really discovering a new antibacterial and having that kind of impact? Or
I think it's the first one rather than the second one. We know that it's more of a serendipity if we landed up on a new antibiotic. And actually, there are much more issues that come forward if the new antibiotic is found like that, because there's this whole thing of who owns the new medicine that's been found, because it's a crowdfunded platform. What country has the rights to develop it? So I'm not sure if we really our goal is not to achieve a new antibiotic, but it's to create a mass movement of awareness and to educate people on the effects of or maybe it's more like how difficult the drug finding is, and if it can be conveyed to the larger audience, we'll encourage a new generation of scientists.
Right, because that's what I was unclear about, how much it was about education and how much about fighting an antibiotic. But you're saying it's primarily about education, but you're also interested primarily in low and middle income countries, but this is pretty expensive for low and middle income countries. So how are you going to get it to them? Who's going to pay?
So, I guess the whole equation would then be on who sees success in this thing. So would it be World Health Organization who's interested in taking this forward and distributing these kits? But yeah, like if it has to really reach out to Africa, Asia, it has to be through a global organization who sees value in this thing.
And it has to be a global organization with money, and WHO doesn't have any. So who might it be? Have you thought of that?
There are few options that I see in my own head. So any charitable organization like Gates Foundation or something who is actually working for decreasing number of antibacterial infections can be one of them. There can be educational institutes. That's what I was trying to point out. If there is value in this being an education tool, then I think the educational institute will provide money. And each of these countries has a substantial amount of educational funds that are available for
Do you think it's a bit of a problem that you've confused in a sense? Is it about antibiotics or is it about science? Does that confuse customers, do you think?
No. I think it's very clear. Like it's creating a new movement of, it's just creating it's using one topic to create a whole new movement of global science.
Right. Any other questions from the panel?
Just building on that, so once you've developed this network of schools and motivated young people in schools and groups, what can you do with that network? Because that seems like an interesting network to engage with.
I think citizen science also has other things that creating a whole new database of plants that are growing can create new ways of looking at what grows where and how can natural biodiversity be prevented from destroying it. So these are the kind of things that can build onto a network that's already created.
Good. Well, thank you very much. So next it's Ethan Tan with the Eco Dream.
Good afternoon, ladies and gentlemen, fellow dragons. I'm Ethan, and this is Eco Dreams, from slums to eco cities. I'm the project founder of this, Eco Dream, and this runs under Boost Venture Group, which is a social enterprise being set up by a group of university students with the passion to empower the communities. So moving on, I'll give on some facts and figures about the area that we are working in. So every year, there are more than 10 million people being added into the urban society within the sub-Saharan region. But however, 2/3 of them end up in informal settlements. And this is a troubling factor as most of them do not reach the age of 55. Many of times, this is because of the diseases that they are exposed to within the informal settlements, such as cholera, diarrhea, and typhoid, along with many others. So, and one of the main contributing factors to this widespread of diseases is the poor sanitation facilities that they have access to. So, lack of access within these sanitation facilities, they are being forced to resort to open defecation, which is resulting them to encouraging the widespread of diseases due to the improper solution and handling of their waste product. So leading on to this, moving on to some quotes where being given by social workers on the grounds where they said that they experience multiple essential arrangements, such as that they lack in the area in which they have no knowledge within the importance of sanitation. And this adds a huge burden towards healthcare costs because a lot of times within the slums, people only visit the hospitals when they are towards the late stage complication of their illness, which adds on to the healthcare costs of the nation. So working in Kenya, so what is the problem? So how do we address this? This shows a compilation of the companies, their donations towards charity sectors. And this actually illustrates and reflects the point that within charity and social organization itself, they are being segregated towards different categories, different segments, which reflects that they work independently, they work in silos, and in this case, that they lack the synergy within different projects that they are working in. So they are all pursuing their own individual objective, individual project. However, and research has shown that the amount of aid going into areas, going to Africa, going to third world countries, have a negative correlation towards their economic growth. So, why is this so? And with this, I would like to propose a concept to change this norm, to propose and to change this problem. Like what Awalu, the head of Nigeria Red Cross, have put it beautifully, to harness our resources and to deliver a solution that can help people in need. So our solution, we envision a network of infrastructure to being implemented in a slum area, within a specific area, to actually address health issues holistically. So how do we do this? We build on the concept of being powered by clean energy to deliver economical and business viable solution towards the area that we are working on. So apart from delivering this, we strive to aim to apply this concept, not just in Africa, not just in Kenya, but throughout the world, in Asia, in South America, in whichever place that needs it. So in that sense, we are using modular design, which brings in the adaptability, the flexibility of each concept that we are working on. However, the foundation, the core remains the same, which is being powered by bioenergy. And of course, this is a large-scale project, so it's a phased implementation. We do not implement the whole project on its own, but on different stages. However, at each point, at each stage in time, we need to ensure that this is being addressed, the problem that we are targeting it at, as well as the resources that we are using is fully utilized. So how does this work? So building on, we will provide a core bioenergy center, which is provided by Enersys company, which is a startup based in France, using microalgae. So using that, of course, we collect waste, organic waste. And further on, letting the algae feed on the waste to produce energy, which is water and biomass. These are the two very essential resources that allows the sustainability of life. So building on from that, we separate water and biomass and distribute the energy towards the community, which addresses different needs. And of course, this is just the core foundation of it. And building on from this, we deliver more value. How do we deliver more value? By addressing health issues in these slums areas. So for example, free public toilet can be connected to this core foundation where you provide proper sanitation services to the slum areas, to the slum dwellers who lack in proper sanitation facilities. And this addresses the core problem of what I've illustrated previously on how the lack of proper sanitation actually contributes to the widespread of diseases. So through this, it actually addresses one of the specific problems in the slum areas that we are looking at. However, being a modular design, we are looking at putting different processes in place, such as with public toilets, it actually contributes to the organic waste that we are collecting, and it produces energy. So being a phased implementation concept, we do not incorporate all the infrastructure yet, due to cost, due to various other factors. The energy produced can be given back to the community in terms of biocans, where in slums, most of them uses this energy for cooking, to cook their meals. So this can be contributed back to the community. And further on, when there's more funding, when the expertise are right, when the time is right, we can bring in different projects, different initiatives from different charities, different organizations into our concept, into our project to build on as a modular design where it constantly grows. And at each point in time, it's addressing a particular need, it's giving value to the community, as well as at the same time, it's providing a holistic support towards the whole network that we are building. So given the flexibility of it and the aim of it of not just applying in just one area itself, being a modular concept, it allows us to shift, mix and match different ideas, different projects into the concept by actually allowing us to design the concept specifically to the area that we hope to implement it in. So of course, there are risks and challenges towards this concept that we hope to implement. Firstly, the technical expertise that we need in terms of implementing it. And I've spoken to Imperial Environmental Policy Department with Dr. Jeremy Woods, where he gave me advice on how to actually tackle some of the specific technical expertise that I'm facing. And along with that, I'm hoping to work with Energy Futures Lab, as well as CHF, which is an international organization that we have been working with for more than a year. They are based in Kenya, and they are providing specific local knowledge towards our implementation. And further on, biotechnology. Of course, there's risk that the technology that we are using may not work in the environment that we are hoping intended to, because this is a new technology discovered by NSys, which is a startup based in France. And they have been using this technology only in test labs and not yet on a commercial basis. So that may run a risk of it being not suitable. Of course, we are working currently closely with them to ensure that the implementation of it fits the intended environment that we are hoping to achieve. And of course, the scale of the project, it may be large, but at the same time, it's being a phased implementation, as well as we need to ensure the relevance of such a large-scale project. So at the same time, I'm hoping to gain some relevant advice from Global Health Innovation in terms of ensuring that it is relevant to the point of what we are implementing. Of course, and our group, Boost Venture Group, where we'll be the team leading this project on a long-term basis to ensure the implementation of it. And lastly, the legal aspect of it. Working in a slum area, working in a different area, there are legal aspects such as you need approvals from government. So as I'm speaking now, Langata Youth Network, which is a local organization in Kenya, they are speaking to the local authorities to gain their support, to gain their confirmation in terms of working within the area that we are working in, as well as getting them on board on our project. So lastly, by connecting different charities together, we hope to create a greater impact, not just to the communities, but to the 800 million people that we are hoping to impact with. And of course, this will help improve their lives, their aspirations, and to actually eradicate health issues that they are facing, not just in Kenya, not just in Africa, but in India, but in South America, and throughout the world. And lastly, we hope to improve their quality of life and allowing them to pursue their dreams. Thank you.
Thank you. Paul.
It's a great idea, but I've seen it before.
Okay.
What differentiates EcoDream from, for example, LuWatt, which is a comparable product and service system?
I would say that the differences it makes entails in the area that we are working on, in terms of the core foundation that we are building on, which is the technology that actually provides different forms of energy, not just energy, but water, and in the sense that it's a holistic approach towards targeting a specific area. But at the same time, this actually allows us to expand. It's a continually growing project, a concept that it adds on whenever there's a need of it to actually address the problem and to actually provide a holistic solution to it.
It's no different from LuWatt.
So, in that sense, from what I've gathered and from what I've known, within the current area that I'm working in, which is Kibera in Kenya, they lack this facility, they lack this area of specialty that we are working on. Where when I was on the ground looking at how they actually gather energy, how they manage their sanitation, it's always working in silo. They are always working independently. So that is why I hope to actually connect all of them together to produce.
Did you know about LuWatt?
Not about LuWatt.
Is it doing well?
Very well. But I think, I mean, to me, for a successful business to take off, or any successful venture, you've got to see what the competition is before you develop your own thing. And I think that what you've got here is identical to LuWatt, except that theirs is a non-water using combustible toilet. But the whole business model is identical to this.
Actually, I would like to add on to the point that ours is not just based on toilet. it's a concept. I'm using toilet because it's a direct illustration of how the concept will work. But at the same time, going back to the slides where the collection of organic waste do not just comes from toilet waste, organic waste, but in terms of recycling, in terms of other aspects of project. So this is not just based on water sanitation, but I've used the idea of water sanitation to illustrate the concept. In terms of, this can be modified in terms of being a clinic, being a recycling area, being whichever need that is needed in the intended slum area. You can apply the project that is needed. So in this sense, I've used free public toilets, but at the same time, you can collect waste from maybe waste management where people are gathering around slum areas to sweep up the waste around the vicinity. And put this waste into the microalgae system and let it feed on, with the microalgae feeding on them and running through and producing energy and water, which is the two most essential life.
Okay, Inga.
Yeah.
Just a question about your relationship with the French is this their slide? The
Enesis.
Yeah. So if I was working for Enesis, I'd be happy because you're promoting that
Yeah.
product before it's even ready. Do you have an idea of when that product will be ready?
They're actually ready in the sense that they're in terms of their finalizing, commercializing phase, and it's being endorsed by the French government. And at the same time, the 5,000 pounds that I hope to get, would go towards pilot projecting this concept, incorporating their technology. But this does not go directly to them. It's just a collaboration with us and their partners in terms of they are providing the microalgae technology while we are using it as a concept to implement it.
Catherine.
So yeah, my question was around, it seems like a very ambitious project. So if you won £5,000 today, what would you specifically do that would drive forward the next phase of the opportunity? So you've kind of answered that, but maybe you can go into a bit more detail.
To be more specific, we have actually identified our pilot site in Kibera, which is Ketawara village, a population of around 70,000 people. So we have analyzed the site as in they do not have comprehensive water sanitation facilities. Waste management is very poor in that area. Diseases, widespread diseases are high. And we figured that this is a place where we can start this project. And currently, within that village itself, they do not use bioenergy. In terms of, they use basic stuff like woodwork, as well as basic AD, aqueous digestion concept, but they do not have current technologies such as microalgae. And this 5,000 pounds goes towards starting this pilot project, implementing the concept, and testing if we are able to create the synergy that we hope to create by incorporating different ideas, not just free public toilets, but recycling waste, as well as different facilities such as clinics, if the area requires a clinic, or even a school, or even, I'll say, a community center, whichever that fits the area.
Okay, not more than 10 words. Who will pay?
The government, CSR, companies, NGOs, with whom are looking at changing the areas that we are working
more
on.
than 10 words. That's good. Well, thank you very much. Let me just tell you, I work a lot in Dhaka in Bangladesh, where 30% of the 20 million people live in slums, and within a few years it's estimated to be 50% of 30 million. So you've got a big market there. Thank you.
Thank you.
Right, carrying on the tradition of clever acronyms, we've now got Nicholas Killis with Daphne, which is the diagnostic platform for heterogeneous and numerous biomarker inputs. Wow.
Hello everyone, as said, my name is Nicholas Kililis, and I'm here today to present to you a new and innovative technology, which I like to call Daphne, that stands for Diagnostic Platform for Heterogeneous and Numerous Biomarker Inputs. I am working on this technology as part of my PhD project, on which I'm supervised by Professor Paul Fremont in the Department of Medicine and Dr. Karen Polizzi, an expert on bioancestral design. What you see here on this first slide is a graph where I have placed technologies that can be used to detect biomarker molecules in biological samples for assessing medical conditions. In the past few decades, there was innovation in biotechnologies brought to the market a large number of small, portable, and affordable technologies which can be used by people to monitor their health. For example, the blood glucose meter is the one that is being used to measure the concentration of glucose in the blood, glucose being the biomarker here, in order to help them plan their meals and medication. And this test is taking place at the comfort of their own house, which is something that significantly raised the standard of care for these people. So imagine if we could design devices like that for many other diseases. For cancer, for example, wouldn't that be amazing? However, there is a small problem here. Diseases like cancer have complex diagnostic profiles. In order to make a reliable diagnosis for cancer patient, you need to get 2, 3, 4, or even 5 biomarkers within the same sample. And this is something that is not currently possible. This is something that's only currently possible in hospitals and with a lot of money. So it's not with expensive equipment. So this is not available to the many. So what can we do about this? Well, my colleagues and I in the lab were working on a platform technology which can be used to detect multiple inputs to develop biosensing devices, medical diagnostic devices. This technology, which we like to call Daphne, integrates many inputs to come out with an answer to complex questions, and it will be able to do this at an affordable price. So this small paper disc that you see here is actually what the diagnostic device will look like. And this contains all the necessary components that you need to carry out the test. Embedded in this paper disc is the hardware for the device in the form of E. coli cell extract, and the software for the device in the form of RNA aptamer sensors. So as soon as you have one of these devices at hand, all you need to do is add the biological sample, let's say a drop of blood, and this activates the device. Then the device computes and senses the biomarker molecules there and gives the response in the form of a color chain indicative of the disease state. As I previously mentioned, Daphne devices will be able for multiple input processing, and what I mean by that is that they will be able to take multiple inputs to deliver a more reliable diagnosis. The innovation which allows, the technical innovation which allows this to happen in this mRNA platform here, which uses RNA aptamer sensors which detect biomarkers in the biological sample. As soon one of these devices, with the switches, binds a biomarker, it changes its conformation so it gives access to the ribosome to produce a protein. In a Daphne device, there will be a number of these sensors working in parallel to integrate many inputs together. Also, the fact that this technology is based on RNA aptamer sensors, it allows us to detect protein, metabolite, RNA, peptide, and antibody biomarkers within the same sample. And as far as I know, please correct me if I'm wrong, I don't know of any other such point-of-care technology which is able to do this currently in the market. So we strongly believe that this technology will encourage biotechnologists to build devices to target new diseases that it was not possible to do with existing technology. The second very important innovation of this Daphne devices is the ultra-low cost of production. So in this table here, I compare the price of producing the cost of production for one Daphne sensor in the lab, which is from 2 to 4 cents per sensor, and compare it to the most widely used point-of-care technology currently in the market, which is antibody-based sensors and PCR-based tests. So as you can see, Daphne compares very favorably to established technologies in the market right now. The third innovation, the third technical innovation, is the one that allows Daphne to be used at the point of care. The sensor undergoes a treatment which makes it available for out-of-the-lab deployment. Also, this same treatment allows it to be stable for more than a year at room temperature without any loss of performance. And as you can understand, this will lead to much lower cost of transportation and storage for diagnostic devices, medical diagnostic devices. Importantly, the results are delivered to the user in in the form of a color change. So this is a simple yes or no answer to the user of the device. However, if the user chooses, he can use the camera phone of his phone to quantitatively measure, follow the results of the test. And what this means is that you can have continuous monitoring and upload this straight away to an online medical logbook. And what this means is that you can have continuous monitoring of one's health by him and his doctor at all times very easily. So, taking into account all of these advanced characteristics, advanced features that these devices will have, I strongly believe that this technology can have a very dynamic entrance in 3 distinct markets. The first market which this technology can enter is the one of healthcare providers and developed countries. The fact that it can deliver rapid test results at the point of care and more reliable, accurate diagnosis means that it's expected to expedite medical decision-making by professionals, and thus increase the efficiency in the system and giving more efficient allocation of resources within hospitals, for example. The second area in which this technology can have a very important effect is the one that provides the toolkits for humanitarian aid workers in developing countries. The fact that the design of these technologies allows it to be used by non-specialized personnel in combination with the extra low cost of production means that this technology can make up, at least to a certain extent, for the lack of infrastructure and medical personnel at this site. The third market in which this technology, I believe, it could have a very, very strong and very successful entrance is the one of home personal diagnostics. Its ability to provide personalized, evidence-based care at an affordable price, it would drive educated consumers in developed countries to use the technology to monitor their health and to change their habits into living a more healthy lifestyles. So, given the opportunity by this institute to carry out further work on this technology, I will use this opportunity to produce a prototype device for this sensor. Firstly, what I will do is I'll try to establish, because I'm a scientist, I will try to establish a partnership with a medical practitioner that has practical experience into being in the hospital with diagnostics. And together, we will try to identify a target medical condition which will be the most suitable first example of these biosensors. Then I will head back in the lab and build a prototype device using the sensors that are needed to detect this particular disease and try to make an analytical assessment of its characteristics. Finishing, I would like to thank the team, which without their help, none of this work will be possible. And say a big thanks to all of you for your attention, for bearing with me.
Inga.
Just got 2 little technical questions. Some of the cost associated and the pain with managing samples for testing is in the preparation of the samples. So you can't use just blood drawn straight out. Do you need any sample prep?
Well, sorry.
Just answer the question.
Yeah, don't worry.
Okay. So I guess for this, I will rely on products that are already in the market, like these printers that take blood. Mm-hmm. So we can see from there how could use other people products that are already in the market to account for that, because actually that's what happens with antibody industry, which they use this antibody-based test like the pregnancy test or that's for urine, but yeah. So
you can copy those others.
I'm not going to copy. I'm just going to cooperate. It won't be into this particular company.
And if you can do multiplex sampling per each circle, and you only have one color change, how does that work? Or do you have multiple circles?
No. It's something that I didn't explain very well. Within the same circle, there will be a number of these mRNA switches that I've shown before, and each one can specifically recognize one particular biomarker in the sample. But advanced assisted biology have allowed us to build like logic gates as the ones you have in computers in the form of DNA and genetic parts. So the actual one circle will compute So you have this biomarker, but you don't have this. So this means that thing. Or like we have 3 out of 4, so we compute like we have 3 out of 4 and giving out a response like in a single color change whether this the disease state is as such. So the color change will always be there like as a single output.
Catherine.
Thanks for a good presentation. So in terms of the point of care mass market, it's kind of quite a busy space. There's lots of people trying to do this, and what I understand from your presentation is your differentiation is the multiplexing of the different types of potential components.
Yes.
So just thinking about point of care, what do you mean by point of care? Is it 5-minute time to get a result, or is it 4 hours? Because obviously that will feed into your application where it can change clinical practice. So where are you going with that? What's your thoughts?
So a change in color at the current state, it takes from 20 minutes to 60 minutes. So depending on the application, you will tune your device to be, and of course the layers of complexity will add to the time. But I hope that as this technology will develop, it will actually take less time to the components will allow it to happen much faster. For example, if we use enzymes or other proteins and stuff like that to amplify and make like an exponential increase in the output of the device, it was something that it will come as the product is developing to make it actually IM for like from 20 minutes like to 40 minutes max. Like it won't be something that would take in overnight or something like that. It would be something that it would be on the spot, either on the bedside of the patient at the hospital, or whether you're using it at home and you take the test, you make your coffee and breakfast and come back and say, okay, I'm missing these nutrients today.
Okay. Paul.
I don't think I've got any questions.
Let me, because have you any idea what sort of condition you're going to test for? Because I imagine someone that's sitting home saying, wonder if I've got colon cancer. You know, boom, 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 sitting there, oh yes, I've got it. I mean, I can't quite think what the condition would be you might be testing for.
The condition.
Yeah, I mean, is it cancer? As you mentioned cancer, is it malaria? What is it?
So this is a platform technology.
Yeah.
So I provide the technology for this to happen. For example, let's say prostate cancer, right? So the poster boy for is like PSA antigen.
Yes, exactly.
It's like the poster boy for cancer. Like if you, from what I know, like if you detect the PSA antigen, like there's 70% possibility that you have cancer, right? So and they actually go ahead with the operation. But their papers and their reviews would say that if you are able to detect up to 5 biomarkers, this possibility, the percentage of certainty, so no possibility, certainty goes from 70% to 96%.
Right.
So that's like a very significant like increase in the, like for people that go unnecessary for operations. So it is something that.
But do these biomarkers exist? Because I mean, PSA is a classic example that confuses everybody. Yes. I mean, I know someone who has tattooed on his chest, don't ever do my PSA, because you end up with all sorts of people thinking they've got diseases that they haven't got and that isn't going. You know, the whole business of false positives and false negatives. I mean, you say either it changes colour or it doesn't change colour, but it doesn't work that way.
That's what actually I'm trying to
get beyond, yeah.
To get beyond. If you notice at some point when I was explaining the technology, was using RNA aptamers to detect not only. So currently the market is mainly relying on like protein biomarkers, right? And that's the most important. That's what is being detected. So being able to detect all these other chemicals, like together with antibodies, together with RNA, that's in many cancer, in blood for many cancer patients, this mRNA, this lead structure that. Or like metabolites, that will significantly increase.
But do these other biomarkers exist, is my point. I think you're kind of way ahead of the medical science here, aren't you? I mean, you've got the technology, but the doctors haven't managed to come up with the biomarkers.
There are in the literature a lot of biomarkers like that, so I'm offering the technology. So I guess as the medicine is progressing, we'll be finding more and more biomarkers for this. For example, like you can do like the genomic test, which say for breast cancer. So technology is evolving, so we need.
All right, so you've got the technology. Now you need the doctors to develop the.
Yeah, that's why I need to contact a medical practitioner to tell me what would be the most.
Well, I noticed you had 2 weeks for doing that. That's right. Well, fantastic! What a great idea. Thank you very much. Right, now we're on to Cafe Communique, which already exists. I've seen it on the web. by Jing Yuan. I'm sure I've not pronounced your name correctly, but you're probably used to that.
So hello everyone, my name is Jing and I'm a final year medical student. Today I'm here to talk to you about Café Communique, which is an online magazine for grassroots discussion into global health. So you might be wondering, Café Communique, bit of an odd name. You know, what's in a name? Well, it's actually a pun on Café Philosophique, which was founded by the philosopher Marc Sauté. So his intention was to create a grassroots forum for discussion into philosophy. And this was open to anyone, regardless of their background or their experience. And in many ways, Café Communique, or CC, tries to mirror that movement. So as most of you will know, global health is bigger than just clinical medicine. You know, it draws together many disparate fields, all under the banner of international health. And we wanted to highlight that to the general public because they're not actually aware of this. Similarly as well, at the time when I started this project, I really wanted to get more involved in the research in all aspects of global health, but there were few opportunities for me. And on speaking to my colleagues as well, there were few opportunities for them. It seemed that global health was confined to academia and peer-reviewed journals. It wasn't really accessible to people such as myself or to the general public. They weren't aware of the issues. So that was one of the key points that we were trying to address. Similarly as well, we wanted to highlight the importance of things like journalism and creative writing. So we very much wanted to create a platform that was a creative outlet for all these students, doctors, academics, to kind of write in this, and packaged in this kind of format, in an engaging and journalistic kind of manner. So those were kind of the ambitions of the project at the beginning. So we had this idea, we decided to create a platform, and we started with 3 co-founders, myself, a girl called Fumida Manon, and a man called Anas Nader. And we're all Imperial medical students. Now, Fumida was doing a global health BSc at the time at Imperial, and Anas has a background in scientific communication. So he had a lot of experience with designing platforms, and he built all the websites that you see today. And I've written here that we followed lean startup principles. So what do I mean by this? Well, we didn't want to create something that was super complicated, super complex, cost a lot of money, that no one wanted. We wanted to see, are people actually interested in what we're proposing? So we started out with a very simple platform, all HTML static pages. That was, very labor-intensive, required a lot of input from our part, from the co-founders. So that was our starting point. We wanted to show that there was a proof of principle, a proof of concept that this could actually work. Because we were working by 2 assumptions here. The first assumption was that the general public actually wanted to read about this kind of material, all on the same place, all in this kind of format. The second one, which is the big assumption, is that there are people out there who actually want to write in this kind of manner. Obviously people want to get published in The Lancet and in PLOS, but do they actually want to write for a magazine? And especially at my level, do students want to do that? So we wanted to test that, and we decided to recruit students from the university mailing list, and we also started our social media as well. So here is our Twitter, and here is our Facebook. And what we found was that there was actually, phenomenal interest from the students. They all came to us and said, "Oh, this is a great idea. I'd love to write," and we were very encouraged by that in the early days. And secondly, as well, for the social media, we found that there was a lot of organic growth. Now, I'm not an expert in social media in the slightest. The first time I actually went on Twitter was to create this account. But we saw that it was just slowly rising, and with not too much input from our part. So it was really encouraging results, and we were really pleased by that in the early days. So we then moved on to the second iteration. Obviously, everything was manual, everything was very high-intensity input. We showed that there was an appetite for accessible global health. And we decided, naturally, to create an automated platform for users to submit content, for editors to edit the content. And we also, in doing so, we were able to expand our team. We were able to scale. And we had a lot more writers then and a lot more editors that we trained up. And we were receiving a broad range of topics, everything from mobile health to medical anthropology. And this is just a screenshot of some of the articles that we had quite recently. And we were getting authors from across the world, so we developed all sorts of partnerships, which I'll touch on a bit later. And from those, we were getting authors, students, doctors, academics, as you would expect. We were even getting, entrepreneurs and CEOs writing as well. For example, a notable one being Simon Berry, who founded Cola Life, came onto the platform to write a counter-argument for an article that he'd seen. So it was really interesting, the number of people that we were reaching. And also as well, Alessandro Demau, who is Australian, the founder of NCD Free. He regularly writes for us. And in terms of automation, so we created an editor space, which you'll see on the left, to oversee the articles and just to keep track of everything. Similarly, we integrated a blogging platform to allow authors to easily submit and write content on the platform itself. So we took a lot of burden from ourselves, the co-founders, and we were able to scale more effectively as a result of this. So partnerships. We spent a lot of time developing a variety of interesting partnerships with a whole variety of organizations. And I like to split them up into publicity, financial, and content. So in terms of publicity, we partnered with many prestigious organizations, such as the RSM, to publicize the opportunity to their audience. Similarly as well, global health institutions like the one at Imperial, like the one at the University of Liverpool, are very much the life force by which we get authors coming through. In addition as well, of course, we involve the student organizations, and they're very interested. So organizations such as Medicine want to collaborate with us to create a paper-based publication as well. And they certainly have the distribution channels for which to do so. So that's something that we're tentatively exploring at this stage. In terms of finances, a lot of people have contributed some money. So for example, the CoLab Center, and also Wesleyan as well. So we've quite recently actually established quite an interesting business arrangement in which they are planning to fund us. But also as well, they'll be synergizing with our social media and also as well using our content for their own publicity material for their customer base. And finally, one of the recent partnerships that we established was with the University of Aberdeen. So what we established was a mechanism in which all the essays, all the coursework that are submitted on their global health and management course will then be sent to Café Communique, processed, edited, and then published on our platform. And the notion for that is that there's so much great writing that takes place within these institutions that just gets a coursework mark and then gets forgotten, never to see the light of day. So we really wanted to put a spotlight on that, and that's something that we're going to be trialing in the next month or so. And that's similarly with the Australian Medical Student Association, which have a publication called Vector, and they're hoping to translate some of their articles onto our platform to enter new audiences. So, of course, you want to see the data. You want to see how well this is doing. So in terms of performance, this just shows our traffic since our inception, which was about 6 to 9 months ago. And we're getting 45,000 hits, thousands of users coming on. And I want to draw your attention to the pages, the session number, and also to the duration. And these are just averages. So the average user, browses about 7 pages. He spends about 4 minutes. He or she spends 4 minutes viewing these articles. So this is just an indication. Obviously, it's not precise, that there is good engagement with the platform as well. We also have a 40% return rate on our platform, which shows. And I think that demonstrates to us that there is a certain loyalty with our fan base, and that's reflected with our social media engagement. That's what we see, that's what we observe. Now, we have quite a relatively high, in my opinion, bounce rate as well, which means that people who jump onto the platform then leave the platform, they don't explore. And the reason for that is because we're not smartphone optimized, and that's something that we're looking to develop in the future. So, what are our next steps? Well, firstly, we want to do a complete redesign. We want to make it slick. So you'll have seen the platform. It looks okay, but we want to make it look even better. And part of that is building a new brand, building a new user interface. And we're already speaking to several specialists in the area, freelancers that really want to get involved with that aspect. And obviously, the internet is a notoriously superficial place. They judge things on look a lot of the time. So that's something that we want to bring up to speed. Secondly, as well, we want to build to scale. So we want to do some more back-end engineering. So at the moment, I mentioned that we're automated, but we're not bespoke. The software does not fit in specifically with our mechanism. And finally, we want to expand our partnerships and also hire a specific social media manager to come in. So that completes my presentation. If you Feel free to visit the website, and if you have any questions, please let me know.
I was pleased to hear you're going to be translating from the Australian. I don't understand Australian. How many Twitter followers have you got?
So we currently have about 130, but we're very Twitter naive.
Right. Catherine?
Just thinking back to sort of the global health impact that you want to have with this initiative, I think it's really great. You're obviously building momentum. What's your ultimate measure of success for this project?
Okay, so our comparison is always that the MIT have a technology review, Harvard have a business review. Why is there no global health review, or at least not a significant one that people want to engage with? That's, I think, our ultimate goal. And I think it all starts from actually building the audience. So a lot of the things we've talked about are online, but offline, our offline work is just as important as our online work. A lot of our success has come from the partnerships that we've formed, and at the moment we want to develop those further. So we started off with quite basic things like mailing lists, and now we're at the stage where we're negotiating with big organizations like Wesleyan, big institutions like the University of Aberdeen, establishing more and more profound partnerships, and who knows where that's going to take us next. For example, we actually collaborate with the IGHI. I've worked with Josie in terms of using some of the public engagement articles that the IGHI publishes onto our own platform, and we show really good interest and really good engagement. So that definitely, I think, shapes the kind of direction that we want to move into. And I think ultimately it's all about the audience. Building a big audience is what we're aiming for.
Have you thought about commercial sponsorship and advertising?
Yes. So I think the Wesleyan deal that we've got going on is kind of a hybrid of that. It's not quite a proper business deal, but it is a sponsorship deal with specific arrangements in place that are both value generating for us and value generating for Wesleyan. Now, in the future, when the audience gets bigger, hopefully, then we'll be able to have more pull with that, and especially if we have a paper-based publication. So as I mentioned, we're collaborating with medicine, and we've been doing this for a while to get a paper-based publication, and from that we'll be able to get a lot more income as a result.
But your costs will go up with paper though, won't they?
Well, at the moment our overheads are very low.
Yeah.
I previously worked at another global health, international affairs publication, so I've got a fair grasp of what the numbers are. And for a publication round, it only costs like 1,000 or 2,000, because our starting point is quite small. We're realistic in our goals, and we develop stepwise. We want to just get it out to the different branches of medicine to begin with. So that's only a distribution of, say, a couple of thousand, and then see where that goes, see what the interest is, and take it from there.
That was going to be my question.
Right. Actually, it struck me. I mean, I went and looked at I think it looks very impressive. It didn't seem to me like you had all that much build. I mean, looking at your data, you seem to have spikes, but not a sort of steady build.
Yes. So I think that's kind of that kind of reflects our own our input at the moment. So at the moment it's all about scalability. Those spikes are where we send out a newsletter. That spike is because everyone kind of remembers it again, but we don't actually have the infrastructure for a mailing list as of yet, and that's one of the things that we hope to build on. So like the mailing list, for example, the mobile platform. Those are things that we're trying to build on. There has been a slow baseline rise, but I think it's kind of confounded because in the early days we put in a lot of effort to publicize. Right. Now we've kind of let it grow, see where it's going. But I think you'll see a small an increase in trend, but with more input from us in terms of increasing our social media and increasing our mailing list publicity, I think you'll see that it will rise a lot more.
And what's your editorial process? If someone sends an article saying, AIDS is clearly nothing to do with HIV or something like that, is that fine, or are we going to stop that? Or?
So within reason, we've actually had debates about certain articles. For example, we caused a bit of tension with Cola Life when we published an article where they basically said, is this a good arrangement? And Simon Berry came on and said, hold on, I don't agree with this at all. And he wrote a counter argument for us. So I think, within reason, so long as it's not completely unreasonable, we will publish it, and we'll always invite, we're always open to the counter argument for that. So yeah.
So what's your long-term business model? You just expect to go on raising sponsorship steadily, or?
So with things like the online magazines, there are many ways of doing it. I think the majority of them follow the business model.
But what are you going to do?
Well, I think we're going to try and follow a similar model, in which there are organisations that do shared articles. So these organisations put on shared content that they want publicised, and that they get advertised on our platform. So you'll see that on many things like BuzzFeeds and like these different like Fast Company and all these different websites do it. And also as well the sponsorship, we seem to be getting a lot of interest from these different organisations that want to help us out, that like the idea that we're engaging the students right at the grassroots level, and they want to contribute. And like I said, overheads are really low at the moment.
So you're going to be the Harvard Business Review of Global Health, that's your vision.
Fingers crossed.
How many people here have seen Cafe Communicate? There's an audience here, you'll probably be boosted by the end of today.
Yeah,
Thank you very
great.
much.
Thank you very much.
Right, now we're on to Christopher Payne and Hanny Marcus, smart handheld instruments for microsurgery.
Good afternoon, ladies and gentlemen, and thank you for inviting us to speak today. And today we're pitching an idea for a smart handheld instrument for the most difficult type of brain surgery, microneurosurgery. What I hope to do is spend the first few minutes discussing the problem of microneurosurgery, and then hand over to Chris to describe the solution that we proposed, which is this novel handheld instrument. But first, a little history. So some of you may recognize this chap as Robert Liston, one of the most famous British surgeons in our history, who practiced in the 1800s in an era before anesthesia and before antisepsis, where the fastest surgeons were the best surgeons. And no one was faster than Bobby Liston. He performed, in his most famous case, an amputation of the leg in just over 2 minutes, which is the amount of time it takes me to prep a patient. And it's famous because, firstly, the patient died, which was not uncommon at the time, from gangrene. Secondly, the assistant who was holding the patient down, fingers were chopped off and also died from gangrene. And thirdly, that when wielding the knife, Robert Liston went through the coattails of a distinguished visiting professor who was so scared he died from shock there and then. So this is the only reported case in history with a 300% mortality. And I should point out he went to UCL rather than Imperial, this chap. Now, there is a serious point here, which is that all surgery is inherently risky. And my own field of brain surgery is perhaps the most risky. And I think there are a couple of reasons for this. The first is that the brain has an incredibly complex structure. Some would say the most complex in the universe, myself included. And so it's easy to get lost if you're not precise. The second reason is that the brain tissue is extremely unforgiving. So if you're not delicate, it's very easy to injure brain tissue. And unfortunately, because the brain has so little capacity for regeneration, if that happens, patients wake up with disability. It causes severe emotional burden to the family and severe financial cost to the state. So the corollary is that interventions which make brain surgery more precise and more delicate improve patient outcomes. Now, over the last decade, there's been a tremendous amount of work on improving precision of surgery, particularly brain surgery, with image guidance. But actually, improving how delicate surgeons are has received very little attention and is the focus of our work today. So we're interested in making surgeons more careful in how they handle tissue. And knowing how much force to exert is actually quite tricky. Clearly, too much force injures brain tissue, but you do need to provide sufficient force to carry out a maneuver effectively. And that balance is something that's gained by surgeons over their training, over decades of training. Unfortunately, often you only know if you've exerted too much force if a patient wakes up with a disability, and then it's too late. So irreversible injury, unfortunately, is one of the ways that we learn as surgeons. So we were interested in helping to improve patient safety by reducing the forces that were exerted by surgeons. Surprisingly, until 2 years ago when we started really, no one had measured the force exerted by brain surgeons when performing microneurosurgery, ever. So the first thing we did was extremely simple. We just measured how much forces surgeons used. And we found two things. The first is that surgeons exerted very little force comparatively when performing brain surgery, an order of magnitude less than when operating on the abdomen or the bowels. The second is that, and somewhat unsurprisingly, when errors happened and patients were at risk, you often found that the forces spiked. And of course, the forces that you exert depend on what operation you're performing, the dissection maneuver that you're performing, who the surgeon is. But broadly speaking, we found a threshold, that red line, below which patients were fairly safe and above which patients were at risk. Not always coming to harm, but at risk. So we thought very simply that surgeons could be alerted if they were exerting too much force and causing risk to patients. Now, the first, the most famous robot in brain surgery is this NeuroArm robot, which is an impressive piece of kit, and I've had the privilege of seeing it in Calgary. It's very complex, very large, very expensive, and there's only one of it in the world. But it's a telesurgical platform where the surgeon sits behind a console and maneuvers things, and the robot recreates those actions faithfully. And it allows you to incorporate this force limits. The problem is that there's only one of it, and we couldn't see this being disseminated into the wider surgical community. So Chris and I thought that perhaps there was a way to incorporate that kind of force limit into handheld instruments that would perhaps be more easy to disseminate. And I think at that point I'll hand over.
So we've opted to essentially augment existing surgical instrumentation rather than develop a new robot altogether. We've taken a blunt dissector and made it smart. So as the surgeon manipulates tissue whilst they're performing the surgery, they exert forces on the tissue, and we've instrumented our dissector to sense these very subtle variations in force as the surgery is performed. We then read this into a computer system and compare to a threshold, which is, as Hani was mentioning earlier, the point at which injury might occur to the brain. And if the surgeon is applying too much force, then the device vibrates. There is this vibrotactile feedback which indicates to the surgeon that they should apply less force and perform the surgery more delicately. So we know at this point that there is a point at which a certain force level will cause trauma to the brain. But what does the brain surgeon actually feel as they're performing the surgery? What are the perceptual factors that influences how much force they perceive? And of course, it's inherently subjective. If you were a brain surgeon, the forces that you would perceive whilst you were performing the surgery would be very different to the person sat next to you. They would be different if you were tired or if you'd drunk lots of coffee. And in fact, there are many psychophysical conditions in which our tactile perception of force might be altered. So in order to validate our device, we had to ask the question, what are the scenarios in surgery in which the surgeon might accidentally, inadvertently apply too much force? So the first one that we looked at was tissue retraction. In some types of neurosurgery, the surgeon will have to retract or move tissue out of the way with one hand whilst they perform another task with the other hand. And we noticed that as the surgeons retract tissue, they inadvertently apply more and more force, which we think is similar to this boiling frog scenario. For those of you who don't know, there is apparently, if you put a frog into a reservoir of water and gradually heat it up, the frog has no perception that the temperature is increasing until it's too late. And we think it's the same situation with tissue retraction. As you exert more and more force, the surgeon's completely unaware of this until inadvertent, irreversible brain injury occurs. So the other scenario is what we've called the high force, low force situation, in which the surgeon, in some cases, might have to apply a lot of force at the very beginning of the surgical procedure. For example, retracting muscle to get access to the spine. And it turns out when you apply a lot of force, it completely throws your tactile perception. And it essentially makes the surgeon heavy-handed, which is obviously not an ideal case for performing very delicate procedure after the initial retraction exercise. So we designed a couple of studies to look at these two particular psychophysical conditions, as well as perform a cadaveric study on an actual human brain in an actual microdissection task. And in all cases, we showed that the device improves force application. It means that the users were applying less force throughout these different scenarios. So to conclude, we developed a microsurgical device which limits the amount of force that is applied during surgery using vibration-based vibrotactile feedback. And it's in the context of developing robotic technology or smart instruments that are much more compact and much more lower cost compared to these sort of master-slave systems such as the NeuroArm or the Da Vinci, which are astronomically expensive. In fact, they're in the sort of region of £1 or £2 million, whereas the devices that we're proposing can be made very, very low cost. They're very simple, and they're also intrinsically robust, because if they fail, it doesn't cause injury to the patient, unlike if one of these master-slave systems should fail. So we feel that we've developed a device that could deliver benefit to the patient, but that could also be clinically translated. Thank you for your time. Paul.
I think my understanding of your product is a bit impaired by my feelings of squeamishness with some of those images. The frog was probably the best of them all. But what I don't understand is it vibrates when there's too much pressure being exerted. Well, isn't that too late?
It's actually at a threshold beyond which certain patients are sort of at risk. So if you look at the forces exerted by surgeons, it tends to climb steadily, and injury occurs at forces which are actually quite high. So we picked a threshold which was at the beginning of that.
You can set, yeah, exactly. You can set that lower than the actual point at which.
So with the instrument, can I have a second question? With the instrument, for example, set the threshold very low when you're going into the brain, set it higher when you're going into muscle around the spine?
Yeah.
Is it possible to, with one instrument, set it to a different?
Yeah, I mean, the more likely scenario is between different instruments. So a blunt dissector and a sharp dissector would have different levels, because sharp dissector would apply more pressure, and the thresholds are actually lower. In fact, that's what the plot that we showed earlier, it's a different level. For actually retracting the muscle, I mean, that's the sort of scenario, that's a lot of force, a completely different order of magnitude, in fact. And the point was that it skews your tactile perception when you apply a lot of
heavy.
force. Yeah, you start heavy, and then you become heavy-handed. You can't do delicate things as delicately.
Yes.
And that was that scenario. But yeah, between different instruments, you could definitely set the threshold.
Do the surgeons you talk to recognise the problem? As in, there must be intra-surgeon variations, so do most of the surgeons look at those graphs and say, that's those other people, I'm the most experienced?
It's difficult because you'll never meet a bad brain surgeon. Apparently they're all amazing. They're always above average, so it's hard.
Actually, I had a friend who was married to one who said, I only do the easy stuff.
Exception. So the truthful answer is actually, I think most surgeons probably wouldn't recognise that they exert too much force. And equally, if you look 10 years ago, the biggest advance in neurosurgery has been image guidance, which is now gold standard. Every patient undergoing tumour surgery or complex surgery has image guidance. And 10 years ago, any surgeon worth his salt would have said it's a waste of time because a good surgeon knows his anatomy and doesn't need to rely on technology. And over 10 years, that's completely changed. I don't think it's, speaking as someone who's happy to admit that I have, things that I'm not very good at, I think it's sort of surgeon comfort is patient safety. I don't think there's room for heroics of doing it with one eye closed, standing up for 12 hours and with an arm behind your back. I think you should make things as easy and as safe as possible.
Catherine?
Just wondering what the IP position is on this.
Yeah, so I guess we'd already spoken to Imperial Innovations.
Correct answer.
And that's where it is.
Okay, but you're confident you've got a position around that? I guess that's the key question.
What about the business model generally? You didn't talk about that at all.
Yeah, I mean, I guess the first step that we would like to take, our ambition, I guess, would be to get this into a patient, into an actual human case, which is obviously quite a big step, which involves developing an instrument that could be sterilized and is robust enough to do that. We think that it is intrinsically robust and it would be straightforward enough to do that compared to some of these other robotic systems which you'd have to predict all of the ways in which they might fail, whereas we know there's only a handful of ways and we can mitigate against them. So we think it is possible. Then I guess it's a case of attracting interest from surgical device manufacturers.
But I mean, if this was a drug, you'd have to go through an enormously complex process of showing its effectiveness and such like. You're going to have to do something similar, aren't you?
Yes.
I don't think that will be. I think it's actually comparatively easy with this device compared to other surgical devices because it's essentially exactly like a normal instrument with one exception, which is that it provides this tactile feedback if you exert too much force. And we know in the studies that Chris was talking about, these preclinical studies, we know that in these very well, randomized preclinical studies, it does seem to improve performance. So I can foresee it being relatively easy to perform at least safety studies in humans relatively quickly. In terms of the longer-term business case, we've talked about this before, neurosurgery is somewhat niche, but there are two important things to consider. The first is that much more money is spent per neurosurgical case than in other surgical fields. And the second is that although we're pitching it in neurosurgery, which is obviously my own area of interest, the principle applies to many surgical fields which involve microsurgery, plastics, ENT, for example.
In your blurb, you suggest it's primarily for trainee surgeons rather than experienced surgeons. Is that just you being sort of polite?
I would draw the analogy, as I did before, with image guidance, which is that when it was initially pitched, surgeons were persuaded to use it primarily for junior surgeons, and now every surgeon uses it. I would say maybe the same thing, but don't tell anyone.
Good. Well, thank you very much. Okay, we're on to our last one, Photo Voice app development.
Good afternoon, ladies and gentlemen, my esteemed dragons. Today we're going to be talking to you...
Don't suck up to us.
I'm standing in the middle of it. Cool. We are the Photo Voice app team, and today we're going to be telling you about an application and a concept that we've devised that we believe and envisage completely revamping the way global health research is done. So today we're going to be talking you through a couple of things. We're going to start by talking about the current research method known as Photo Voice that exists and the problems that we have with it. Then we'll introduce our Photo Voice app solution, tell you a little bit about it. We'll talk about the product and the business model behind the application, and most importantly, we'll go through the future applications that this technology can have.
So, over the last century, the world of politics, media, and global health has been revolutionized by photography. These kind of photos have really changed the way that we understand the world and stories around us. But let's just think for a second, how are these photos actually helping the individuals and their global health needs? So when you take a photo, and when someone takes a photo of a problem, in many ways a photo like this is the photographer deciding what the problem is of that person. And what we want to do is stop allowing other people to decide what global health problems people might be experiencing, but by giving cameras to people and letting them tell you their own story, you understand their position and their health problems from their own perspective. So Photo Voice is a research method that's already in use, where you give cameras to people and you let them explain through the power of photography what their problems are. So it's often done with minority communities, and by giving them the cameras and exhibiting the photos, or having group discussions, or publishing them in museums, you can allow people to understand their global health problems. It's been used in a variety of fields, but it's mainly done by epidemiologists, anthropologists, scientists, and artists. And it's really important in breaking stigma and discrimination in all health problems. So I'm going to talk you through two case studies that have been published very recently, and giving an example of how A, Photo Voice can help break stigma and discrimination with all types of problems and diseases, and B, how it can change the actual world that we live in. So this is a study that was done last year, where cameras were given to freed sex slaves and people who've been trafficked from North Africa into Europe. And they were asked to take photos of the problems that they've experienced with terms of discrimination and stigma and fitting into society. So what does this photo mean to you? Probably not very much. It might just be a blurry truck. But actually, this photo was taken by a participant from that study, and it's the truck that she was trafficked in and escaped from. And this just illustrates how the power of photography can really transcend language and cultural barriers. So many of the participants are often unable to talk about their problem because they might be illiterate, they might not be able to participate in surveys and research problems, and by giving them cameras they can really explain their problems through photography. Furthermore, if you're experiencing trauma or some kind of awful situation, it's very hard to describe your problem, and it's actually quite empowering to use a camera. This is the second case study that I want to talk you through. It was a study that was published, obviously, in a high-profile medical journal recently, and it was giving cameras to disabled people in Turkey. And what the point of the study was to use these photos and obviously have group discussions to get over the stigma and the discrimination that people were experiencing, but also to use the photos to change public opinion by publishing them in a museum and build a map to show exactly where these photos were coming from. It's quite a slow process, but this map was built, and as well as the publication, it effectively named and shamed buildings that had poor disability access. Since this was published, we've been in close contact with Özge Kaman. I met her at the World Health Summit in Berlin a few months ago, and since then she's been helping us design our product and improve the service. She said that about half of the buildings that were identified in her study have actually now redeveloped to allow disabled access to be much more important. And this shows how photo voice can actually really change the physical environment that people live in. Okay, but are there any problems with this camera method? I think this is the only room where I've seen more cameras than maybe iPhones. But I was going to say how many people have a camera phone. I'd imagine many of you. This camera method is actually quite expensive. It's old-fashioned, and translating the results into real, tangible products is quite slow. Because of the expensive nature of the research method, it's limited in its access. It's thousands of pounds per participant for a study often, and if you don't have a printer or a computer to download the photos on, you're limited in that sense too. So what we want to do is solve this problem. This is to illustrate the rapid rate by which the world can change. So in less than 10 years, we've gone from a world where very, very few people would use their camera phone or their phone to illustrate problems. But now, we've got a world where people are taking photos of the most menial things and the most important things. And this is a way that we can imagine harnessing the growth in technology and the huge amount of people who use camera phones. The fastest growing smartphone markets are in Africa, Southeast Asia, and Latin America, and the price of smartphones are dropping phenomenally. So I've identified some problems. I'm now going to talk through a couple of the solutions. So by providing a mobile app where this camera method is translated down into a service on your phone, it would actually be extremely cheap for participants to use it, and we think that we can envisage, instead of just hundreds or thousands of photo voice projects happening per year, possibly millions. It's modern, so young people love using their phones and new technologies, and often as photo voice projects are done with minority groups, this is a key area of people that would be interested. Uploading photos is instant, and we're going to increase access. If we can bring the price down, then more and more photo voice projects will be done, and we can enter different areas of global health. Okay, so this is an idea of what the service will be providing is. So harnessing the power of a phone, we'll be managing the relationship between the researcher and the participant. They can discuss when they're going to have their meetings and how the photos are affecting them and arrange all that kind of stuff. And then using the camera phone powers, the participants can add description to their photos if they want to. Then harnessing that power, what we would provide is a service to the researchers where we can analyze the photos, store them online, and in fact, we can actually build very simple maps very quickly using geotagging, so making the process much more efficient. This is a basic mock-up of how the app might look. So it's really key that we'd have straight-to-camera mode, so very easy to use. You then add a few comments, and you might have, for example, 100 characters to describe the problem, 100 characters to describe your solution or suggestion, and then you might have a profile. Now, each research project will be different, so some will want to have confidentiality, others will want to have lots of information and use the network to discuss problems between users. So this could be adjustable. What you'd then do is, using your photos, you'd tag them or send them towards a certain study. So this is an example of how, for example, we could do a disability access study in London. And in fact, in the future applications, once we've improved the service a bit more, we could if there was a research project looking at barriers to collection of medicine in Africa, you could take a photo of the problem and instantly upload it and say, this is stopping me from getting to work, I'm missing lots of hours, it's taking me ages, I can't get my medicine. How many of you would actually write an email or write a complaint or get it stamped and all the right approval when you have a problem like this? If you were a participant in a research study, you could instantly direct your problem with a photo. Again, this is a basic mock-up of how the app might look. So you have a profile, you'd be pinpointing using geotagging on the maps where your photos are coming from, so you can build areas to show hotspots of problems, and this is how you'd add data and description to each photo.
Now, the beauty of the Photo Voice app is that we're leveraging the power of mobile phone technology, and we're bringing down the cost of current photo voice research methods. Together, both of these combine to create a whole new world of opportunities. And this brings us to our market entry strategy. So what are we going to use the money for? We're going to start by building a minimum viable product, which we'll use with a couple of organizations that we've already built relationships with, and we'll get them to really try the product out using pilot studies. Throughout this process, we'll constantly be learning from this process, improving upon the product as well. The next step from here is to learn how to analyze the data and create software and a platform whereby we can manage this data and make it easily accessible and understandable for the researchers as well. Once we've done this, the opportunities for global growth are endless. It's important to note as well that currently global health researchers spend a huge amount of money investing in these photo voice research methods, which are old-fashioned and inefficient. And what we're doing is modernizing this research method and using technology to bring it into the 21st century.
So this is a list of all the projects that Photo Voice has done this year, and by creating an app we believe that we can cover all these areas and more. So we've already been in discussion with 3 potential pilot studies that we're going to do once we've created the app. The first will be with the Migrant Resource Centre in the UK, discussing migrant health beliefs and health behaviours. The second will be about patient experience and support within the NHS. And the third will be a follow-up study of the disability access in Turkey that we mentioned earlier. Once we've got a refined app that's being used globally by all Photo Voice researchers, we want to spread the net of Photo Voice wider. Even though it has the possibility to change global health widely, we believe that even you can use it to harness change within your own community. For example, it could be used by an NHS psychiatric support group as a method of facilitating discussion as part of therapy. Another way that we can see it being used is by industry in design and innovation to target areas that are in need of development. Given all this, we believe that Photo Voice has the power to revolutionise global health research as we know it. Thank you.
Could you talk a bit about competition? So
Of course, yeah.
If you start doing well, then what's to stop other people copying?
So we've looked at an app called Fix My Street. Don't know if any of you are aware of it. So what they
in New York.
Fix My Street. I think it's
Everywhere, isn't it?
It's run by the government here, local councils. You send photos of graffiti, broken windows, that kind of stuff, and they, it's effectively what I was saying about having this long process of writing a complaint. But there's no one using it in global health or for health problems. So in terms of the competition, yes, there's lots of people with smartphones, and we're not saying that every single person with a smartphone is going to be using it, but what we do know is that this research method is being used at the moment, right now, and they're limited by the number of participants just due to the cost. If we can provide a service where people are using smartphones as well as cameras, or instead of cameras, we can widely...
So why can't people just use the Fix My Street technology?
Because when you send a photo on Fix My Street, it gets sent to the local council, so I don't know what...
Can't you just send it somewhere else?
Not at the moment. People have suggested, why don't you use Twitter? Why don't you use Instagram? You can take a photo, you can send it, but what do we, the people who use Twitter and Instagram are not going to be able to analyze the photos. What we want to do is provide a service where we can analyze the photos and also, really importantly, keep them confidential.
Yeah. Catherine?
Just thinking about, maybe I missed it, but who annotates the photos then? Because if you're saying you're working with people that might not be that literate, is it the worker who's working with them that annotates photos, or how?
Yep, so there's different ideas that we've had. Okay. So one of them would be where as soon as you take a photo, you answer 3 short survey questions designed by the researcher. And this could be generic, so describe the problem, describe the solution, or it could be very specific. Or you could have a group, so you also have group discussions. That's a very important part of this method, where you meet up and you could have the researchers asking the questions and getting people to annotate the photos retrospectively. Can I? I've just noticed...
You want to ask us a question?
Well, if I can. We've just noticed our software developer team has got here late from an exam. And he...
Good excuse.
Yeah, fair enough. He's got a prototype on his phone. And if you wanted to see it, we could show it to you.
We can do that during the drinks reception.
During the drinks reception is fine.
Paul, do you have any questions?
I don't think so, no.
So, if I understand it, the customers are going to be the researchers.
Not quite. So the researchers would be paying maybe for a commission to use our service.
Well, that's right. They're the people who are paying, in a sense.
The researchers, yes.
And how? So you've got data on how much they're paying at the moment, and how much cheaper you think yours would be.
We do, yes.
So you know there's a market.
We've used Qualtrics as we had a business student who's used Qualtrics to analyse by doing surveys to different researchers to work out what their costs are. And we've seen their data. We know that in the UK they spend about £500,000 a year on photo voice projects, but very little impact is happening because of them.
So how much could you do it for, do you think?
Well,
because you're going to do it the same thing, only better.
Definitely, it's definitely going to be better. We're going to be providing the service for cheaper and for less money.
So that's win-win then. So how much is it going to be? So a minute people are spending £500,000. What are you going to do it for?
Sorry, that's the cost overall.
Yeah.
So we could do a study, for example, where if we have different business models. One would be that you'd pay the researchers would pay to use their app for 100 patients for a month, and that would cost X amount. Or we could do it where you put it on per patient basis. So you're paying £1 per person who's participating. At the moment, we know that the app development is our main cost. We've been working with some students who are happy to show stuff later on, but we need to get some kind of well-designed back end where all the data can be analysed. We built a front end where you can take photos and send it to people, and we built a front end for the researchers where you can look at the map where photos are coming in. But we need to have storage space and app development costs.
So last question: is your ultimate aim to be millionaires, give a voice to the marginalised, or both?
So at the moment, I want this to happen. That's my most important thing. We've been in contact with Ozgi Kaman, who wrote the Lancet paper on it, and she...
And if you don't get the money, can you still make it happen?
This is going to happen. If we don't make it, if you guys don't help us to make it happen, it's going to happen. It's an inefficient process which is going to have to change.
Okay, thank you. Okay, well we've come to the end, so we judges are now going to sneak off and make our final decision. But before we go, I want to observe what tremendous kind of creativity and energy. I think it's really fun. And also I would say, I've kind of learned over the years that if you really want to make something happen on a big scale, somehow you've got to think business. Otherwise it's very tricky. So I think this is really admirable what the people here are doing.
Yes.
So we'll be as kind as we can when we make our judgments, but remember you've got to make yours too. So we're off.
Thank you, Richard. That you were planned to have 40 minutes, now you only have 25 minutes to...
We'll manage.
Okay, good. So let's give a round of applause to the dragons and also see them later. So last year that we had our winner, that Gabrielle, and she's going to share with us her experience of participating in this competition, and also her journey of using the winning money and actually to turn some of her ideas into practice. So let's welcome Gabrielle.
Hello everyone, I've had a really fantastic time listening to all of this year's competitors. It's been a really fascinating afternoon, and thank you so much for inviting me back to speak this year. Just to talk a bit about kind of moving forward from what's happened since last year when I was kind of in this place presenting research I've been doing on schistosomiasis. And I've stood here and I talked about diagnostics of schistosomiasis and kind of the existing techniques and a novel technique that I was looking at called LAMP. But before I continue, just a little bit about schistosomiasis. So schistosomiasis is a neglected tropical disease, and I was looking particularly at S. mansoni, which is an intestinal form of the disease. And most of its symptoms are very non-specific, and therefore they're really difficult to attribute solely to schistosomiasis. And currently, most of the kind of control programs are through mass drug administration with praziquantel. And in 2011, 243 million people were treated with praziquantel for schistosomiasis. As mass drug administration continues, and infection intensities are beginning to drop, and areas with lower infection intensities are beginning to be targeted. And so good diagnostic tests are essential for the evaluation of these treatment programs. So far, the existing diagnostic techniques have not been very accurate at detecting the low-intensity infections. Existing techniques include kind of catechats, which is here, which is microscopy looking at stool and detecting individual eggs in the stool. And they're great because you can, as well as looking at just for schistosomiasis, you can also look at other neglected tropical diseases. And then you've got here antigen tests that exist, which can have quite high kind of startup costs, but they can only look at S. mansoni, and they're not really kind of available to use for other things. And these existing techniques have not been accurate in low-intensity areas, and there's been a massive discordance between the results, which I presented last year. So my research I'd been doing last year was looking at the discordance between the existing results and trying to find a third diagnostic test to try and evaluate that discordance and to try and find a diagnostic adaptation. So I pledged this last year. So I said that I would look at LAMP and I would try and improve its field applicability and look at it as a novel diagnostic technique. And LAMP is an isothermal DNA amplification technique. So you can amplify DNA in less than an hour at a single temperature, which makes it great in the field. So I've been trying to investigate and improve LAMP's field applicability and accuracy. And as I left last year, I had to look at accuracy, cost, and ease of use of the tests. And I had a lot of ideas, and I left with many ideas, many questions, and a lot of possibilities. I wasn't really sure how I was going to make this happen. I talked about looking at making DNA extraction easier from the crude samples we've been using, and maybe looking at trying to find a test to try and, we had a lot of problems with contamination, to try and find maybe a one-unit test to try and decrease the contamination that we'd been having. So the challenge was, how was I going to achieve this? And I had a lot of ideas, and I thought about potentially going to Japan to talk to the scientists that have had a lot of success with LAMP out there. And they've been really making massive strides forwards to detect that. And whilst that would have been very interesting and I would have learnt a lot, I really wanted to find a way that I could leverage what I had. So leverage the £5,000 that I'd won here and try and turn it into something more. So I thought about finding an existing technology to combine what they'd already achieved and then think about what I want to achieve and what we had achieved and try and make something what we hoped to achieve and try and find a better diagnostic tool. And the reason why I kind of eventually landed on that is how I wanted to proceed was because I realised that actually the IGHI prize had given me something more than just the £5,000. As an undergraduate student, I'm a final year medical student. It had given me credibility. So having a group like the IGHI believe in what I was trying to achieve made other people believe in it too. I had legitimacy. So as an undergraduate student, I was suddenly able to engage with world-class innovators who couldn't wait to hear about what I told the IGHI when I was presenting. And it also gave me financial resource. So it gave me allowed me to buy my place at the table. It gave me an opening bid and allowed me to leverage what I had into an even greater potential. So in speaking to different people and throughout the year, I eventually met DNAE, which are an imperial-based spin-off, which was started by Professor Tumazu. And I've just got some quotes here. Oh, well done. I've got some quotes here from their website just about kind of what they do. And they provide these kind of microchip diagnostic tests which use the LAMP assay, so what I kind of developed in the lab, and you can put it on the microchip, plug it in to a computer, and it can give you your results. And the computer heats up the chip, and it's all done in one system. As it's all contained in one system, they have a lot less problems with contamination that we've been having in the lab, but also allows them to be used as a point-of-care test. On top of which, they have multiple wells in them, so you could use it to test for beyond just kind of S. mansoni, which is what I've been working on, but also potentially develop them to look at kind of other forms of schistosomiasis, like hematobium, which is a urinary form of schistosomiasis, as well as other neglected tropical diseases that we'd looked at. And so really, what I wanted to use the money for was a proof of concept. So I've been discussing with them and working together with them to try and develop our assay to put on their chip, to see if we can get a small number of prototype samples to prove the concept. And when I first met them, I was a little worried. I had no idea how much these cost and how, because they've been using them throughout several different industries and different places, and I kind of really felt like it was, didn't really know how to proceed. But with the backing from IGHI and what I'd spoken about here, they were keen to speak to me further and to learn more about kind of what we'd achieved and what we hoped to achieve. And so we're really working on this to try and kind of develop it further and to develop this kind of point of care field-based test. So since last year, I've also kind of finished working on the lab and finished looking at the LAMP samples that I'd ran. And when I presented last year, I had kind of very much kind of crude results that we'd had at the time. And also been working on a new set of primers so that with the test, we'd also be able to look at several types of schistosomiasis. And while this has all been happening, there's been a massive boost and increase in interest in diagnostics for neglected tropical diseases. So the Bill and Melinda Gates Foundation have announced that they're going to invest heavily in new diagnostics and industry collaborations via their PATH program. So that's exactly what we've been looking at. We're just a little ahead of them. So now they're coming in to looking at kind of what we've been doing, and so that's kind of great for moving forward once we've proved the concept. So where from here? So we're starting to look at developing a point-of-care diagnostic test that would be field applicable, that can be developed to test NTDs beyond just S. mansoni. So really, from this, the possibilities are endless. I'd just really like to thank the IGHI for all their support and help. Without them, it really wouldn't have been possible. And to the support I've had from Dr. Gow and Dr. Lamberton, who are my supervisors. So thank you.
Perhaps you can stay on. Let's take 2 brief questions from the audience, because there will be 3 of them will be the winners for today, and they probably want to know that how they are going to use the money wisely. Any questions? Well, let me ask you one question then. That what the IGHI competition has made you to do things otherwise not possible.
As for me, I think for me it opened up a world for me. So it made me, it kind of allowed me to approach people, to discuss things, and really to look at things in terms of kind of leveraging what I had in order to kind of create a greater thing from what I'd achieved, and to really look at things more in a kind of a business or how to proceed things. So what I really started off when I was here last year was with just the lab results I'd had from looking at the tests.
So for the potential winner, what is your advice, what to do and what not to do?
Well, that's a very difficult question. I think it depends on. I think it's just to keep trying and keep pushing forwards, and even if whether you win or not, you can always make it to the end. I think.
Thank you very much for
coming.
Thank you
so much. Thanks for inviting me back.
You will be more successful in your ongoing journey.
Thank you.
Thank you very much. So now this part of the presentation is almost to its end. And before we actually cast your vote, so each one of you should receive this, your money, your investment. And if you haven't got one, that mark over there, I'll see, yeah, we've got a few more here. So the ballot boxes are all outside. And I hope you do remember the name and order with which all the presentations are made. And just put this in there, and then we will do the counting. So the idea is that the dragons will decide the first prize and the second prize, and you will be deciding, the audience will decide the third prize. So there will be drinks reception at the main entrance of the college entrance downstairs. And before we go down, I would like to pay special thanks to Jo, who's our comms officer at IGHI, and she has put in a lot of time in organizing this event. Jo. Standing over there, Mark, who's our administrator for IGHI, manager for IGHI, and he and Jo been really working together. A lot of the ideas, and also this idea of audience vote is very much from both of them. I thought it was a jolly good idea and a way of engaging the audience. Thank you very much, Mark. So lastly, but not least, let's thank all the finalists as well as all the participants for supporting us for this Students' Challenge competition. And I hope you will spread words. And for next year, there will be more entries, and also I'm sure that we'll try to make this event involving more students, and also maybe that outside Imperial as well. So thank you very much for coming, and enjoy the drinks reception. Thank you.
Nice. Congratulations. Thank you. Where's your surgeon? He's had to rush off to operate on a brain.
He's on call, yeah, he's absolutely.
Well, I hope he doesn't apply too much pressure.
Let's hope not.
Oh yeah, and you get a... Here we are. So take that.
Come on, Chris.
Yeah. So here we are. Look at this. There are two of you, so you'll have to take it turn and turn about. You know, 1 week in one house and then the other.
Guys, if looking this way, hold your certificate up for me.
You should hold this.
Well done.
Good. And look over to me, please. Good. Smile, please.
You want to with all the judges?
Yeah, with all the judges. Here we are. You come into the middle.
Do you want to bounce up and come this side?
Good, close together, guys. And rather than like soldiers,
Yeah,
just turn a slight angle.
There we go. Well done.
Okay, guys. Expo, well done.
So I was wondering, why did you...
I think, well, just because the brain is...
He's got brains just lying around. So it's like sort of...
I know.
Putin and Obama.